Very good morning. Today we discuss about the biotransformation of the drugs. Before we start my lecture, today is the Deepavali and প্রত্যেকে আমার প্রীতি ও শুভেচ্ছা এবং দীপাবলির এই আলোর মধ্য দিয়ে এবং এখন যে সিচুয়েশান কোভিড-19 যে ভাইরাসের কবলে আমরা রয়েছি এবং সেখান থেকে যাতে আমরা মুক্তি পাই এবং সেখান থেকে মুক্তি পেতে গেলে একমাত্র আমাদের ঠাকুরই ভরসা এবং সেখানে আমরা সবাই শক্তির আরাধনা করব। তার সঙ্গে আমাদের দরকার যারা মেডিকেল পার্সন তাদের প্রতি আমাদের শুভেচ্ছা এবং তাদের সবাই বাড়ির প্রতি সবারকে আমার শুভেচ্ছা। এছাড়াও যেটা করতে আমাদের সবচেয়ে বেশি সাহায্য করবে সেটা হচ্ছে ড্রাগ। তো সে ড্রাগ মেটাবলিজম অ্যান্ড হাউ হোয়াট আর দ্য ড্রাগ এফেক্ট অন দ্য করোনা ভাইরাস অ্যান্ড হাউ ইট উইল বি মেটাবলাইজড, this is very important. So you can learn about this topic. I think it will be great helpful for the metabolism of the drug as well as the you can design the drug and you can prevent the any type of disease and the viruses also. So thank you all my listeners. Again I about the biotransformation of drug part 2. Today we discuss about the basic consequence like definition, consequence, type and physics, phase 1 and phase 2, enzyme, induction, inhibition and fast metabolism. Biotransformation means or metabolism means the chemical alteration of the drug in the body. It is need to render non-polar or lipid soluble compound to the polar soluble or lipid insoluble substances or so that they cannot reabsorb in the renal tubules and are excreted. This whole process is called the biotransformation. So simply the non-polar substances can be converted into a polar substance. That means they are the precipitate form, they are not absorbed by the body, they are excreted through urine. Most of the hydrophilic drugs like streptomycin, neostigmine, pancuronium and are the little biotransformed and are large excreted in the unchanged form. So they are not biotransformed. Mechanism which are the metabolized of the drug and essentially foreign substances are developed to protect in the body from the ingested toxin also. The primary site of metabolism other than this the liver, kidney, intestine, lungs and plasma. And biotransformation of drug leads to the following two interesting topic. One is the genobiotics, another is the phytoalexin. What is genobiotics? Substance and the foreign to the body include drug and processed food additives, cosmetic products and environmental pollutant and ergochemicals. These are the chemicals. Study with this chemical, it is called the genobiotics because these are the very toxic to the body and how it will be eliminated by the biotransformation. Another is a phytoalexin or dietary plant toxin. Biotransformation need to detoxification and the protect of the body from the ingested toxin. The chemical alteration of the body, non-polar to polar lipid substances converted into the polar substances. And the main site of action is the liver. First one, the greatest biotransformation of all the things. Another is the kidney, intestine, lungs and the plasma. Drug after the biotransformation, generally three types of biotransform is formed. One is the drug inactivation. That means inactivity of the drug and the active metabolites are rendered inactive or less active. That means when the drug is administered, after its utilization get the therapeutic activity, then it is inactive. Say for example, ibuprofen, paracetamol, lidocaine, chloramphenicol, paracetamol, and its side metabolite, active metabolite is the para-hydroxypropanol. Another group 2, the active drug to achieve active metabolites. In this form, there the drug is having the therapeutic activity and their metabolite also the metabolic activity. And active metabolites are from the active form drug and drug may be found in the partially converted to the one or more active metabolites. And the effects observed and the drug as given below: chloralhydrate and trichloroethanol. To chloralhydrate the original drug and trichloroethanol their metabolite both are active form. That means they get the therapeutic activity. Say for morphine and morphine 6-guanidine, cefotaxime and diacetyl cefotaxime. These are all the active and the metabolite forms, but they give the same therapeutic activity, like digitoxin and digoxin, phenylcysteine, paracetamol, primidone, dimethyloxygen, digoxin, digitoxin, amitriptyline and nortriptyline. Another group C, this is active, inactive drug or prodrug, or in the form active drugs. Activation of the drugs, they ever forward the drugs that are achieved such a and need to convert the body, and one or more active metabolites such as drugs are called the product. And these prodrug are maybe advantageous of the body. So prodrug are the drugs which is just pro means primary, when the drug before that is not therapeutic activity, but after this transformation it is a active product. That's why it is called the product, and product is very important for the research purposes, and there is so many advantages for this product concept. It is a more stable than the original drug and having the better bioavailability. That means their absorption and the bioavailability is much more higher, and other desirable pharmacokinetics, that means ADME of the others drugs is very important, and less side effect and less toxic. These are the prodrug which are the less side effect than the original drug, and toxicity also less. And some drugs are the activated and selected their site of action. Say for example, you remember it very in your competitive examination, levodopa after it is converted to dopamine. Levodopa is a prodrug. It is not active against the Parkinsonism because it is a CNS stimulant, but levodopa cannot cross the blood-brain barrier. But there are active metabolites, dopamine, which is easily cross the blood barrier and control the Parkinsonism. Say for example, enalapril. Enalapril is not active, not reduces the blood pressure, but the enalapril, their metabolites is enalaprilant, which is the active form of, they are getting the pharmacological activity. Methyl dopa, methyl norepinephrine. So the left hand side, all the products, they are having no pharmacological activity, but their right hand side, the active form of metabolites which are having the biological activity. So careful, like acyclovir, which is the antiviral drug, which are not active, but the acyclovir triphosphate, which is the active form of antiviral drugs. So these are the short summary of this active form of drugs. One is the active drug to the inactive, like say for example lidocaine, chloramphenicol, ibuprofen, and the active drugs to the toxin intermetabolites like primidone, dimethyl. So it is a very good interesting feature of this, the active drug. Or but after metabolism there is a formation of through active inactive drug, active metabolite or product, inactivation, and active metabolites after active drug during the action of the get the prolonged transformation. So all are active, but they are converted into a three forms. One is the active to inactive, active to active, and inactive compound converted to active compound. What are the type of biotransformation? Generally we type of biotransformation. Classification of the biotransformation, generally two types. One is the phase 1 non-synthetic or functionalized, and reaction is the functional group. It is generally generated or exposed in the metabolite, may be active or inactive. A functional group is generated, metabolite a active or inactive. Phase 2 synthetic or conjugated, reaction metabolite are mostly active except of few drugs. A glucuronic conjugation, not in the glucuronic, basically the glucuronic acid of morphine and sulfate conjugated, and the minoxidil were active. The endogenous radical is conjugated. Metabolic is usually inactive. So biotransformation, we can see it, the drug after the oral absorption, first one the phase 1, after that it is oxidation, reduction and hydrolysis, or the drug coming from the following phase 1. The drug may be activated, unchanged or most often inactivated. After it get into the phase 2, or the drugs, some drugs directly enter into the phase 2 metabolites, and after phase 2 metabolites they are conjugation product, and this conjugated product drug however usually inactivated, so their conjugated product will be excreted from the by the urine. So they are the phase 1 reaction. Number 1, oxidation, reduction, hydrolysis, cyclization and decyclization. They are the inverse conjugation of drug. It is a phase 1 metabolite with a endogenous generally derived from the carbohydrate amino acid, and to form a polar highly ionized organic acid which is easily excreted urine and bile. Conjugated reaction have been highly energy equivalents. Glucuronide conjugation, acetylation, methylation, sulfate conjugation, glycine conjugation, glutathione conjugation and ribonucleic conjugation. The first one is the oxidation. Oxidation or non-synthetic or phase 1 reaction. Oxidation, the reaction involved in the addition of oxygen or negatively charged radical or moved to the hydrogen positively charged radicals. Oxidation are the most important drug metabolites of reaction. Various oxidative reaction like hydroxylation, oxygenation of carbon, nitrogen and atom, oxygen of O, dealkylation, oxidative deamination. It may be causes the initial insertion and oxygen to form a drug metabolite and produce. So oxidative reaction are the mostly carried out by the group of monooxygenase and the leaves, and it gives the final steps involve the cytochrome P54 happen in the protein NADP cytochrome P450 reductase and the molecular 0 more than 100 cytochrome P54 isoenzyme different in the affinity of various substances. So here we can see there is some cytochrome enzymes that are metabolized by the different compounds basically present in the liver. The cytochrome 3A4 by 5 and carry out the biotransformation of the largest number, near about the 50% of the drug, and in addition the liver and erythromycin and clarithromycin. The CYP2D36, it is the next most important CYP isoform. Actually this is the form of P440 isoform of enzymes. They are metabolized most of the drugs by in the liver, and which is metabolized by the 20% of the drug, including the tricyclic antidepressant and selectively serotonin reuptake inhibitor, arrhythmic drugs and blockers and the opiates. And another number 2, the CYP2C8 by 9 important drugs in the biotransformation. Greater than 50% of the commonly used the drug of the phenytoin, warfarin, which is the narrow and margin, and CYP2C19 metabolite more than 12% use of the drugs. Say for example, rifampicin, carbamazepine. Another is the CYP2E1. If the catalyze the oxidation reaction alcohol and the formation of the minor metabolite of the drugs, and notably the neprotoxin N-acetyl benzaquinone from the paracetamol. And another the reduction, it is the second highest most reaction in the body drug metabolized. The reaction at the covalent covers the oxidation and involve the cytochrome P450 enzymes working in the opposite direction. Alcohol aldehyde quinone are reductase. Drug primarily reduced by the chlorhydrate chloramphenicol and halothane. Next, this is the highest most hydrolysis. This is the cleavage of the drugs molecule by taking up the molecule of a water H2 ester plus H2 ester H, alcohol dehydrogenase, alcohol aldehyde plus alcohol, aqueous aldehyde plus alcohol. Similarly, amides and polyamides and hydrolyzed by the amidase and peptidase. In addition, there are the peroxidase, hydroxylase, which is the detoxified and metabolized from the some drugs generated by the CYP oxygenase. Hydrolysis occurs by the liver, intestine, plasma, and the other tissue. Another is the cyclization, that means cyclic reaction. There are non-cyclic compound is converted to cyclic. This is the form of ring structure from the straight chain compound like proguanil. And then the decyclization, that means the reverse process of the cyclization. This is the opening of the structure, and cyclic drug one we converted to straight chain structure like barbiturate, phenytoin, and this is the generally the minor pathway. Phase 1 reaction, oxidation, this addition of oxygen or other electronegative compound into the radical and to form a hydrogen positively charged. Oxidation is the main process of metabolism. Reduct unstable intermediate, epoxide, oxide, and quinones. Oxidation in the 9 types. Oxidation at nitrogen, say for substrate in the RNH2 in presence of oxygen RNHOH. In case of chlorpheniramine, doapsone, mepiridine. Chlorpheniramine maleate, it is antihistaminic drugs. Dapsone is the antitubercular drug, and mepiridine it is the analgesic drugs. Oxidation at sulfur atom. This is the substrate with oxygen. These are the form. The chlorpromazine and chloramphenicals. Aliphatic hydroxylation. Hydroxylation group added RCH2 CH2. Salicylic acid is to gentisic acid, ibuprofen, tolbutamine, and glutamine by the hydroxyl group added to the drugs. Aromatic amino acid, aromatic group of amino acid in presence of oxygen, it converted into a para-hydroxy amine compound, phenytoin, barbiturate, and paracetamol. Dealkylation of oxidation, these are the substrate in presence of oxygen, they are converted into a RCOH and H2. Generally it is occurred in the phenacetin to paracetamol. And dealkylation of nitrogen atom, RNH2 on presence of oxygen, NH2 and CH2O, amitriptyline to nortriptyline. Dealkylation at sulfur atom, RSHCH3 and oxygen, RSSHCH2, X-methylthioine to thiomercaptone. Oxidation, deamination, RCH2O, RCHONH2, amphetamine. This is a very important component which is acts as a CNS stimulant. And disulfur or parathion to peroxide. Desulfuration, these are the compound. So we can see it, the main enzymes for the oxygenase, the microsomal monooxygenase in the liver, cytochrome P50 and CYP family of the drugs can be. CYP is required NADP and oxygen, and the drug can be metabolized by the 2 types, and the microsomal CYP 450 and UDPGT, non-microsomal flavoprotein oxidase and esterase. Non-microsomal enzymes like mitochondrial enzymes, oxidative deamination of adrenaline, 5-HT, serotonin, and tryptamine. Cytoplasmic hydrolase dehydrogenase alcohol oxidation to aldehyde and acetic acid, plasma oxidase to histamine and genobiotics. These are reduction. This is the second most highest reaction in the Biotransformation, addition of hydrogen positively charged radical or removal of oxygen. That means in that case the positive charge or added or the negative charge is removed from the reaction. It is called the reduction. The microsomal reduction by the monoamino oxidase is needed in NADPH and cytochrome C reductase. Nitro reduction in NO2 RNH2 chloramphenicol to aryl amino acid. Keto reduction cortisone to hydrocortisone. Azure reduction prontosil to sulfonamides. And non-microsomal reduction chloralhydrate trichloroethanol. Hydrolysis, drug is split combining with the water, ester with water, esterase, alcohol with acid. This is very common reaction. Microsomal hydrolase and pethidine to mepiridine acid. All are the very good analgesic drug, pethidine and mepiridine. Non-microsomal hydrolase, esterase and amidase peptide. Atropine to tropic acid. Cyclization reaction, these are the form of ring structure of the straight chain compound, as for example pro reaction, and decyclization reaction, ring structure open, phenytoin and barbiturate. So today I stop here. Next day we discuss about the phase 2 reaction. Before that, thank you for listening the lecture. If you like it, please like me, or if you have any question, write your question in the chat box or in the Google Classroom. Thank you again. Shubho Deepavali.